All right, our final presentation in this MCubed different segment comes from a cube called Integrative Study of Interspecies Disease in Kenya's Diverse Drylands. Team members include faculty, a postdoctoral fellow, and a graduate student from the School of Public Health and the School of Natural Resources and Environment. Here to represent are Professors Johannes Foufopoulos and Rebecca Harden.
Thank you, Valerie. And thank all of you for hanging in until this very last cube. We want to tell you the story of our cube, and it's a story that is one of how the integration of our two fields, very different fields, disease ecology and cultural anthropology, living under one roof at the School of Natural Resources and Environment, was able, with MCubed support, to take root and bloom as a fully fledged collaborative international interdisciplinary project, looking at what a particular pathogen can tell us about social and environmental change and vulnerability in really ecologically diverse and economically diverse drylands in Africa. Drylands where very high populations of wildlife and high diversity of wildlife coexist, as we've long discussed from our own work in other sites around the world, not just in spite of, but because of human systems, which historically have coexisted with the wildlife and indeed been low enough impact to ensure that these species-rich ecosystems are truly modern ones. It's also a story of how going into Kenya as teachers ready to learn and teach with Kenyans and with our own students taught us, in literally a life and death situation, to listen to how locals are defining problems that require research and need more knowledge. In 2010, we were invited by the Graham Institute of Sustainability to teach a field course integrating social and ecological field methods for studying change in these kinds of ecosystems. We were delighted. During the course of our time there, we learned so much about a new constitution that was changing land rights that Kenyans had ratified, commercial agriculture expanding, wildlife disease and management, tourism industries. It was a perfect laboratory for the inequities and dynamism of change in rich, arid ecosystems. We met experts that ranged from the veterinarians at the Kenyan Wildlife Service to international wildlife veterinarians, laboratory and modelers experts looking at disease and public health. Most importantly, a Michigan alumni, Margaret Kinnaird, who was directing the Impala Research Center, a foremost center for field ecology studies, and her colleague with whom she was supervising students at University of Kenya, Jesse Njoka, a professor who runs a center for sustainable dryland systems. We worked with them and learned a lot, including some things we didn't mean to learn. At one point, when my daughter fell ill from an oral typhoid vaccine and became severely dehydrated, she was unconscious and we rushed her to a local hospital in the nearby town of Nanyuki. It's a field researcher's worst nightmare, as I'm sure many of you can imagine. And when that generous, competent team of Kenyan doctors and nurses restored her to health with an IV drip, I was literally on my knees like, how can I ever thank you? And they said, well, actually, can you get Q fever test kits? And I was like, can I get what? This sounds like an alphabet soup. What is Q fever? And they said, well, we don't really know, but we think we might be seeing a lot of it in this region. It looks a lot like brucellosis, but we may be misdiagnosing it, and we think a lot of people don't know it's out there. It could be a silent epidemic. So I went back to the disease guy and said, what do you think? I mean, it looked to us like studies of this were mostly being done on tourists returning from these arid ecosystems or on Princeton students who were being screened before and after their time there. And in fact, we thought if communities are at risk and they don't know about it, maybe there's a gap to fill here. Maybe we can help take this pathogen, learn from it, and teach about it to prevent it in the future. The more we learned about it, the more that seemed like it might be a really important thing to do. I'll let Johannes tell you about that.
Thanks. So we came back to Michigan and we started looking at Q fever. And it turns out this is actually a very interesting disease. It's a little-known pathogen that has a worldwide distribution. You can find it from North America to Africa. And not only that, it actually infects a really broad range of hosts. It can be found in multiple species of wildlife, it can actually be found in several species of livestock, and it infects humans. What it also does is it starts pretty innocuously. The first symptoms are kind of relatively mild, pretty generic, but if left untreated, the disease progresses and becomes devastating infection of the inner organs, the liver, the lungs, and worst of all, the lining of the heart, leading eventually to endocarditis, which invariably is fatal if left untreated. So we realized this is a pretty serious pathogen. At the same time, I learned to my chagrin that this is actually the most infectious pathogen known to man. A single bacterium is enough to start a new infection. And we know that because back in the 50s, 60s, and 70s, Uncle Sam did a lot of research on this bacterium because they realized that this is a great biological weapon. So at this point, we realized, okay, this is something we really need to pay attention to. So one of the questions is, how can you pick up this disease? One way this can happen is by getting your morning milk, pastoralist style. So if conditions are less than sanitary. But the truth is that this doesn't just happen in Africa. This can happen in other parts of the world as well. So as a matter of fact, 2 years ago, there were 2 people who actually got sick with Q fever right here in Washington County because they were drinking unpasteurized milk. So this is actually a pretty global occurrence. Other places where you can actually pick up the disease is places where lots of livestock congregate and you can actually inhale infected dust particles. Places like markets where a lot of animals are brought together. You can also pick it up at places like bomas, as we found out from our old personal experience. So these are places where people put their animals overnight to protect them from lion attacks. So, and if you're particularly unlucky, you can actually get the disease from if you're bitten by one of these beautiful amblyoma ticks, which also transmit the disease. So ultimately, there are multiple ways in which we can pick up the disease. And at that point, we realized, coming back, that we really will need the help of a bona fide epidemiologist. We were very fortunate because MCubed allowed us to start our collaboration with Dr. Joe Eisenberg, who was in the School of Public Health, and he's actually an expert on tropical infectious diseases. Unfortunately, he's not with us today because he actually has to be in Washington, D.C., communicating the results of our research and meeting with colleagues. But it was he who really helped us take this to the next level. And I have to emphasize how difficult it is to do this kind of work, because traditionally you have experts with narrow expertise dealing with one particular issue. But this is an interdisciplinary problem. You need to understand the epidemiological pathways through which the pathogen changes host. You need to understand the ecological drivers underlying these patterns, and you certainly need to understand the social end of things, the social factors that affect transmission in human societies. So we were lucky, with the support of MCubed, to actually be able to do this, to hire a postdoc, and to start addressing hard questions like this one here. How does the pathogen spill around between the different groups of hosts? How do different types of land management, whether you have a conservancy or you have an intensive ranch, affect the transmission of the disease? And we were able to actually hire a postdoc and graduate students and take them in the field and start collecting data. Here's, for example, Annie Wang collecting blood samples from camels. Her take on the whole thing is, if you want to take blood from a camel, don't do it alone. Okay? So never underestimate a desperate camel. So we trained local students to actually do this kind of work, lab and field work, and we're very happy to report our first results. So, for example, which were both interesting and counterintuitive. We found out that against what we were expecting, it turns out that actually Q fever is pretty rare in cattle, which is the most common livestock species in the region. It's more common in sheep and in goats, but most interesting, it's highest in camels. And the reason why this matters is that as the climate in that part of the world changes and it becomes more arid and more variable, humans are looking to camels as a way to actually adapt to altered environmental conditions. And what's happening now is what looks like a really good solution for the future may not be quite as good if the camels are a huge source of Q fever for local societies. So it's this kind of insight and this kind of information that we hope will help people live healthy and productive lives in that part of the world.
Again, the emphasis really stays on the teaching and learning with our colleagues in country. Our field team from last summer, just to give you a brief update, including postdoc Pete Larson in the CAP, we could never have scooped him up as he finished his Kenyan fieldwork on malaria and say, come on over to our field site without MCubed funds. He and Annie worked with Deshawn next to him under the camel head, who's a veterinary student at the College of Veterinary Medicine in Nairobi, local camel herders, KWS authorities, in labs, in the field, in workshops, in partnership with their faculty who've been coming to U of M with us to write big grants for the Gates Foundation, NSF's Emerging Infectious Disease Program. We're all really enthusiastic about where this can go from here, and we're amazed that if you stay close to the ground and look at what people are asking you to look at, and then take that into places where you think people are most vulnerable, you can actually still generate methods and theory that redefine hot, cool, cutting-edge fields. And we find that really exciting. So thank you, MCubed. Thank you also STEM Africa here at Michigan, Sisters Fund from the Women's Studies Center, SNRE. All of you, including the Graham Institute, have helped us get this off the ground and let it take flight. We hope to be at it for years to come, and we appreciate your attention.
Really an impressive set of talks, hasn't it been? And I want to take a moment right now to recognize a wonderful member of our event team who has been the coach for these speakers today. And this is Kelsey Rhodes of TEDx U of M. Kelsey? As you can see, delivering a TED Talk is very different from delivering a traditional academic talk, so I really want to recognize the faculty for stepping out and learning this new genre. Well, if you enjoyed that MCubed segment, please know that there are more great stories to come. This afternoon in the Mendelssohn Theater at 1:30, we'll move into the MCubed Difference Part 2. In just a moment, we will invite you to move over to the Michigan League for the poster session of the century. Over 170 cubes will be available to talk with you one-on-one about their exciting projects. And for an overview of the poster session theme-based divisions, as well as floor plans, you can use your event program. Box lunches are also available there in the league from now until about 1. So you can grab lunch as you're interacting with the poster presenters. You'll have plenty of time for those conversations. Be sure, though, to come back to Mendelssohn Theater, not this space, but Mendelssohn Theater over at the League this afternoon at 1:30 to see the next segment of the MCubed Difference. See you there.